animal department, where we are doing feeding studies in rats with genetically modified ingredients, food, and DNA constructs. So we are now opening a room where the rats are living. They are very well fed. They have a good life here. And here are our scientific helpers. This is a very unique experiment in the sense that it's the first experiment we know of where you have designed it so that you can detect any difference between these rat groups, and then you can go backwards and find out what this difference means in terms of health, for instance, or in terms of malfunctions of the organs or whatever. The background for this is that many places in the world, people and domestic animals are already eating genetically modified food or feed originating in genetically modified plants. In addition to that, the intended use in humans and domestic animals, of course, as you will know, on its way from the soil and to the table or the animal house, a lot of different species and animals will begin and consume of the genetically modified plants, and we don't know anything about what effect this will have on any organism. The situation up till now is best described by the headline of an article in Science by my colleague Jose Domingo, and the headline read, Health Effects of Genetically Modified Food, Many Opinions, but few data. After the rats are sacrificed in the Department of Experimental Animals, the organs have been frozen down at a very low temperature, and then what we want to do is to analyze the organs and see whether our foreign DNA has arrived in those organs, and in what condition the DNA can be found there in the organs. While in some experiments in a German group with mice, this German group, which is led by Dr. Walter Dörfler in Cologne, were demonstrating that some types of foreign DNA were not cleared from the organism, from the mice organisms. It went to the internal organs and were even inserted into the DNA of the mice. If that is the case, if that happens, then it may be the start of a highly unwanted process with regard to health.
It seems like a wide-scale experiment on humans, in view of the fact that genetically modified food has been on the market for 8 years and already eaten by millions of Americans. However, it is an experiment being conducted without test groups. No knowledge can be gained as to whether and in what form our health is affected if one group eats genetically modified food, but a test group is lacking. The entire population is simply subjected to the same potentially harmful substances. A few scientists suspect that there might be a connection to the increase of chronic illnesses and the weakening of the immune system. And the consumer might wonder if he may not have any children if he eats sterile fish in the future. Do we at least know what repercussions this has on our environment? At Purdue University in Indiana, Bill Muir and Rick Howard are performing tests and doing pioneer research work to determine what actually happens when genetically modified fish, soon to be introduced on the market and enter the food chain, mingle with wild fish. For this purpose, they are breeding their own transgenic fish to remain independent of the food industry.
A number of animals have been made transgenic, including animals that are commercially important. One group that has been studied quite a bit in terms of making transgenic individuals are fish, and fish for commercial purposes. So 15, 20 different species, like salmon, like tilapia, like carp, have been made transgenic. And so we had the facilities and also the expertise to investigate the problem in fish, but also to make our approach to answering those questions such that it could be used in pigs, it can be used in cattle, it can be used in any other type of organ, or plant, any type of organism. Because again, it involves general features of biology that's common to any organism. To make the transgenic fish, we get the recently fertilized egg, and usually within 5 minutes of them being fertilized, we bring them over to this room and go through a procedure called microinjection to literally inject into the egg thousands of copies of small segments of DNA. And those segments of DNA include the gene that we're after, in this case the salmon growth hormone gene, as well as a promoter that turns that gene on. And we're inserting that into a tilapia egg that has just been fertilized. So what our student has here is eggs on a small strip with a very thin glass needle that contains the DNA, and he'll put that needle right up to the egg membrane, and then with air pressure shoot the DNA into the egg. And from there on, it's a matter of chance as to what happens. If the segment of DNA happens to be in the right place, then it gets incorporated into the chromosomes of the organism, and we're successful in making a transgenic fish. But that may only happen about 2% or 5% of the time, so you go through many laborious processes of injecting the egg.
Well, you have this extraordinary situation where we're taking human genes and putting them in a fish, and we're mixing and matching the genetic makeup of the entire living kingdom and the whole biotic community. And who's deciding? We're changing the permanent genetic makeup permanently of the entire animal kingdom, and who's deciding? In our Congress here in the United States, throughout legislatures throughout the world, we vote on all these different laws, tax laws and corporate laws. What could be more important than deciding on the permanent genetic future of life on Earth? But we don't vote on that. A very few scientists and regulators and corporations impose that on us. But there's no referendum. There's no elections. And this is one of the fundamental issues, I think, that we have with democracy. Democracy is legislation. Technology is legislation. Technology actually is the basis for almost all major social change. But we don't vote on it. We vote on everything else. But whether it be the nuclear bomb or the automobile, are now taking human genes and putting them into other animals and mixing and matching all the genes down. We don't vote on that. Oh, no. That would let a few corporations, scientists, and regulators decide. I think that's a fundamental question we need to answer about technology today, is that we no longer can let just a few decide these questions that will last for millennia. We need to say technology is legislation. Technology is a law that will determine our future, and we need to vote on that. We need to be able to decide. We need to become informed, and we need to make the choice.
This is at the heart of what Anne Kapuscinski is fighting for single-handedly at the University of Minnesota. Bothered by the fact that the approval process is so secretive, she began to conduct her own experiments. With grants for independent research, she examines the behavior of the transgenic fish bred by Bill Muir and Rick Howard. In an old farm building, she tries to reproduce a simulated ecosystem in many tanks and aquariums in order to conduct experiments resembling a real-life situation.
Oh, there was some sunfish in there. Okay, you guys, time to get some food. They're really lazy compared to trout. Trout and salmon would just be jumping like crazy. But, these are tropical fish, so they're used to taking it easy. I'm teasing.
Her reputation as an independent scientist reaches all the way to Thailand. the industry is especially anxious to capture the Southeastern Asian market with genetically modified tilapia, a popular fish on Asian menus. For this reason, it is pressing the government with applications for approval to be able to sell its transgenic fish earlier than in America.
And the government of Thailand became quite worried because they felt that they were not well equipped to review an application and even know what questions to ask and how to do a risk assessment and be able to make a good decision about whether they should allow the fish into the country. So they told the researchers, please don't even apply formally to introduce these fish because we don't know what to do.
Under great time pressure, Anne is compiling a risk assessment report together with her Thai colleague concerning the possible dangers to the environment should transgenic fish be approved for commercial purposes. For, just as with foodstuffs, the potential consequences to the environment have so far not been thoroughly explored.
One of the things we want to know is if in the future the Thai government approved genetically engineered tilapia, and if they were to escape from the fish farms, and we know they will since the regular farm tilapia have, would that cause more harm, or will it be equal to the possible harm that the farm tilapia that have already escaped are posing?
To explore the possible risk factors, Anne is working with a small, fast-reproducing fish species from Japan. Many thousands of fish are measured, photographed, the eggs counted, and their mating behavior observed. Transgenic male, wild female. Wild male, wild female. 2 males, 1 female. Which one asserts itself? Which offspring are stronger? None of it sounds like quick fame or profit. More like hard work at weekends and overtime.
Last night we put this fish trap in the tank, and the reason for doing that is we want to collect a sample of the mixed population. This is a mixed population because 6 months ago we released some genetically engineered fish into this tank that had contained a wild population of the same species. These are Medaka that come from Japan. That's their native origin. So we imported them from Japan. We're taking advantage of the fact that in these fish a generation is only about 2 months rather than 70 years or 80 years like it would be in a human. And there was enough time for 3 generations, because in this species they reach sexual maturity at 2 months. The purpose of this experiment is to determine if genetically engineered fish will outcompete and mate with wild fish, and if they do that, whether their genes will spread in a wild population or whether they will disappear over a number of generations. And we're interested in that because that's one of the main questions about ecological risk. If genetically engineered fish are being produced in a fish farm, and if they were to escape from the farm, and if they were able to mate, if they were fertile, the question is, they escape, and if they escape into waters where there are wild relatives, what will happen if they interbreed with the wild relatives? And our experiment is designed to test that question.
To this end, Rick Howard and Bill Muir of Purdue University developed a computer model in which they created a population of 60,000 wild creatures into which 60 transgenic individuals penetrate. A catalogue of questions was compiled. For instance, are there certain survival strategies or mating advantages? And are the mixed offspring stronger or weaker? These phenomena are observed and the results recorded. Then, the computer calculates the possible future results.
is to look for mating advantages of transgenic males relative to wild-type males. Transgenic males are larger than wild-type males, and they could have increased mating success because of that. And they could have increased success because either the female prefers to mate with larger males, or that by being larger, they can drive away the smaller wild-type competitors that are around. And as a result of that combined advantage with other males, as well as the female preference, we found that the transgenic males get more than 75% of all the matings. So, for example, one thing that we've also measured is that the young don't survive as well. And the mating advantage of the transgenic males would drive that transgene into the population. It would become more and more transgenic, but the survivorship of the young is less and less through time, resulting in a smaller population size until, quite likely, the population could go extinct.
Genetic engineering, to some extent, is about a 400-year-old mistake. It was this mistake that began with the Cartesian revolution and this idea that life is a machine. Descartes said that, that basically animals are bet machines, that animals are basically machines. And the Cartesians would vivisect cats and dogs, and when they would hear the screams, they would say, aha, this is like the gears shifting in the machine. That's where this noise is coming from. This was a totally mechanistic vision. And if you trace the last 400 years, you can see there's been a certain part of the scientific community, by no means all, that has continued this mechanistic myth, very dangerous myth. So now that they see the entire living world as simply machines, and genes as the software that you can move around, it's a pathology. It's a terrible mistake. It's not a premise. It's a pre-miss. It misses what is unique about living organisms. And yet they continue to try and treat life as machine and engineer it as if it were a machine. And the cruelties of the early vivisectionists are now being repeated by the genetic engineers who are literally changing the makeup of the entire living kingdom based on this pathological mistake of thinking that life is a machine. That's why they believe in genetic engineering. They're engineering life as if they were engineering machines. That's the fundamental mistake of genetic engineering.
As a lawyer and author, Andrew Kimbrell battles his way through all the issues raised by the new genetic technology. He heads an environmental agency in Washington, which vigorously campaigns for food safety.
Literally, legally, as an attorney, I find this very important. For the very first time in history, in the last 20 years, we've defined plants, animals, even humans now, as machines and manufacturers. Under Section 101 of our patent law, that's what you can patent, machines and manufacturers. So we've decided as a government, as a polity in the United States, that a pumpkin, that a beagle, that a human, and a primate, that these are machines and manufacturers, no different than refrigerators or toasters or a new tennis racket. They can be patented and commodified. This is a shocking commodification of life and a shocking philosophical development as well as legal development.
This is a greenhouse that was used for storage, and so 1 year ago, this facility had a lot of things that had been stored over the years, which we had to clear out, and then construct this entire facility to hold the tubs and for the channels. So we have about 150 tons of water in here right now.
Old farm buildings, converted greenhouses, improvisation and inventiveness. This is what risk assessment research looks like conducted by a handful of idealists around the world. One would think that it is the obligation of industry and government-controlling agencies to conduct these tests. But markets and profit are at stake, not animals and food, and most certainly not our environment, that has not only suffered the effect, but may even be in grave jeopardy.
The research that we're doing here and looking at the transgenic mating advantage and so forth is very unique because we know of no other lab in the world that is looking at the success of transgenic individuals and wild-type individuals. And actually, one of the reasons why we began this research in the first place is to set a methodology where industry, where regulators could test organisms and see whether they would be safe if they enter the environment. Ways that transgenic organisms like fish can get into the environment in the first place would most likely be an accidental occurrence, where there would be either an impoundment or a fence area in the ocean that the fish would escape from and then go into the natural population. And every year, thousands and thousands of fish escape from these types of situations. So it's a very common type of event. There was a storm off the coast of Maine a couple of years ago that destroyed some of the enclosures that Atlantic salmon were being farmed in. And in that one storm, 100,000 fish escaped. So they can escape from these situations, and they can escape in great numbers.
There are certainly environmental hazards associated with transgenic animals, and particularly with fish, because they can escape and they're free-ranging after that. It's real hard to find one after they get out, as the salmon farming industry has discovered on its own. In order to protect against the fish either colonizing new habitat or interbreeding with wild fish, what we're doing is developing a fish that is our production line. Fish that will be sold will be sterile, so that they can't reproduce, and they will be all female. And the reason why they're all female is because sterile female salmon tend not to come back from the ocean. They have no reason to come back to the rivers to spawn because they never mature. So they stay out to sea, they feed, they live their lives there, and they die there.
One of the things that I find so curious about the argument of the biotechnology companies that often call themselves life sciences is that when you talk to them about the environmental threats, about all the other threats, they say, don't worry, we are making genetically engineered fish sterile. We're going to make sure they're sterile. By the way, who checks on this? Millions of fish being sterile is a ridiculous enforcement idea. Don't worry, biological pollution of plants. We're going to put a terminator technology in these plants so they'll commit suicide after one growing season. And I find it very strange that a company that calls itself Life Sciences is telling us that their technology only will work if we make all life on Earth sterile. What a terrifying concept. You know, if I was an engineer, and an engineer came to me and said, I've invented a technology about life, but the only problem is we have to sterilize all living things, I'd still go back to the drawing board. You have a failed technology. That's what I'd say. Because that's, he's going to tell you, Alabama, they're going to make it sterile.
In Norway, not only are the effects of foodstuffs on humans and animals being examined, Terje Travik has brought together a group of scientists from numerous different fields to work out a holistic perspective. They include molecular biologists, geneticists, immunologists, ecologists, and most recently, a philosopher.
And those questions are both concerned with the whole ecosystem, disturbances in the ecosystem, by introducing new, foreign, possibly different DNA, and also directly related to changes that may take place in animal organisms and in plant organisms. We are concerned about what we call genetic pollution, and we are concerned about making everybody understand that genetic pollution is something totally different from the chemical pollutions we have
been stupid enough to initiate over the past 50 years or so, because chemicals never replicate themselves. Even a huge chemical pollution will over time get smaller. While for DNA, it may be the other way around, because DNA is self-replicating in principle. So a small pollution may replicate itself to become a huge pollution, in theory. And it's all these different types of risk aspects that we are concerned about contributing to getting answers to. So far it's a lot of questions, no answer.
Just like their American colleagues, the Norwegian team wonders about what effect these new forms of life will have outside the laboratory. Experience gathered with genetically modified farming has shown that there are and will be grave repercussions to the environment. Just as pollen fly and the modified plants drift to unwelcome fields, fish that escape will undo the predictions made by industry and no longer be subject to control. Man is only one link in the food chain. The entire ecosystem is affected. Thomas Burns is a member of Terje Travik's team and sees the matter from an ecological viewpoint.
Yes, I have been to Cuba, and we have some cooperation with a group of scientists from Cuba. And as a very interesting example of a transgenic fish, it has some other traits that was not expected when they modified the genes of the fish. They have found that the tilapia fish, it is growing about twice as fast as the normal tilapia, but as a side effect, or a totally different effect, it also tolerates salt water. And that may be very important in, for example, the further spread of that species. So it also shows that a new transgenic plant or organism, it may suddenly have some other traits that was not expected and maybe not wanted, and just come as a side effect of the modification.
The difference is that we don't put that first generation of crops or animals out onto the market. We observe them. As I said, in our case, we have 5 generations that have been under cultivation where we have been observing these fish, and we've been culling anything that has an unexpected result, something that continues growing fast, or grows too quickly, or gets sick, or whatever it is. We cull those fish, and we only select the ones that don't have those unintended side effects for actual production. By the time these fish are ready to go on the market, they will have been through 6 generations. That's over 15 years of observation. And we're quite confident that there's nothing occurring there that was unexpected.
Back to the United States. For the past 8 years, genetically modified grain has been cultivated as if this were completely normal. Canola, cotton, and soya dominate the market. The plants have been manipulated so that they produce their own insecticides to kill pests. Apart from humans, who of course eat this as well, how does it affect nature? David Amdo, professor of entomology at the University of Minnesota, is attempting to examine just this.
The point is that the BC corn, BC cotton, and even BC potatoes were commercialized before many of the potential effects of these crops on the environment were investigated. So how much gene flow, what kind of non-target effects, whether or not resistance from the target will occur, and how to deal with that. these things were not figured out before the plants were commercialized. And it was as they were commercialized, people were raising these issues, and frankly, what it is it takes a while. It takes a number of years to figure these things out. And the people who made these plants knew that they wanted to get them commercialized as fast as possible. So you run into a problem where the people who are trying to sell these things want to sell them as soon as possible, because the sooner they sell them, the faster they can make their investments back. And at the same time, we need to take the time to evaluate the environmental effects. And so in the United States, the route that's been taken is to allow them to be commercialized and then sort of play a game of trying to chase after it and find out whether or not we have any effects and characterize what they might be. So this is a laboratory of insect ecology. We are interested in various factors affecting insects in the environment. So of the many things we do, we actually work on endangered species problems. There's an endangered species of butterfly nearby that we work on. We also work on non-chemical means of controlling insect pests. And the area that you're probably most interested in is our work associated with the environmental effects of BT maize. Now, the two areas that we're working on there are the way that BT maize may affect the evolution of resistance in the target pest, the European corn borer. And we're conducting a lot of work on that because we have been rearing corn borers in this lab now continuously for probably since about 1948, when corn borers first appeared here. The other things that we've been looking at is how the BT maize may affect the non-target insects. So things that the BT maize isn't supposed to affect, but may affect inadvertently.
I think the importance of monarchs to the ecosystem is a pretty interesting thing to think about. So probably, if monarchs went extinct tomorrow, there probably wouldn't be a big ecological impact. I mean, there are a few parasites that depend on them and some predators, but nothing that's really they aren't what we call a keystone species. They aren't a species that affects huge numbers of other species. And monarchs, because they migrate, depend on habitats in many different parts of North America. So an individual monarch butterfly that emerges in Minnesota or somewhere else in the northern part of its breeding range will migrate through the central part of the United States, through Texas, and into sites in central Mexico, where they spend about 4 or 5 months, and then fly back into the southern part of the United States, where they start another generation of monarchs. So what we're doing is we're trying to figure out the relative impact of the genetically modified crops and all of the other things that might be killing monarchs in their environment.
The monarch butterfly lays its eggs on a weed, the so-called milkweed, that grew on fields. After the industry had developed a supposedly ingenious method of killing all plants, except for the desired useful plants, by the calculated use of a certain herbicide, it took away the butterfly's habitat. An unintentional side effect of gene technology in agriculture.
I don't think that we should think about things solely in terms of either ecological or economic importance. Monarchs are something that has evolved in a very amazing way. They're the only insect that has this long distance and regular migration. And they're interesting and they're beautiful, and they have a right to live just because they've evolved and just because they're there. So I think it's really important for us to think of what we can do, what they need in the habitat, but not necessarily what they can do for us, besides just interest us.
We now know to our experience with corn in the United States, that the biological pollution of these genetically engineered crops is uncontrollable. There's no buffer zone. You can't control the way insects fly or that rainwater will carry it. Any vector can take these new genes and spread them to other crops and to weedy relatives. It's happening. All over the United States can't control it. But the companies are not taking responsibility for that, and they're not being held liable for this biological pollution. In the future, a company like Monsanto is going to go out of business. They're teetering economically anyway. Monsanto's in tremendous economic problems. Think of the billions of dollars already out there in biological pollution costs they're not going to pay. So they're going to be long gone. But meanwhile, we're going to look at our major crops if we're not careful. Corn, soy, cotton, wheat, rice, and they're all going to be polluted, perhaps indefinitely in the future because of these companies' actions which they can never pay for. That's gross corporate irresponsibility.
Now, if you consider a field in Minnesota, and you ask how many species of insects are there in a typical maize field through a growing season, what we could say is that the studies have shown is that there's approximately 700 species of insects that visit maize every year. And so if you think about how many pests there are, there's maybe about 5 to 10 species of pests. So all the rest of those species, the other 695 or so species, would be considered the non-target species. So there are far more non-target species than there are target species. And so when you try to control the target, it's very likely that you're going to affect some of those other species as well.
The industry attempts to destroy 5 insect species that cause losses worth millions. That is understandable. What is more difficult to comprehend is that industry does not seem to care that almost 700 other animal species are also affected. Claudia Strahlen, a Swiss scientist, came to America to join David Ando's team and to study the effect of this poison on these small living creatures. The scientists of Norway and America are just at the beginning of their research. Quite frightening, for the grain is already on the market, and industry is impatiently awaiting approval of transgenic fish.
I'm trying very hard to get the government regulators, the other scientists, and the consumers to all understand that risk assessment has to be done, and it has to be done in a scientifically sound way. And it's complicated work, looking at each kind of genetically engineered fish, figuring out a good methodology to do the risk assessment, and then on a case-by-case basis, figure out what is the most reasonable answer. We're always going to have uncertainty, and the scientists can't get all the answers. That's why the democratic process is so important, because it's society that has to decide what uncertainty are we willing to accept, trade it off with what possible benefits and what possible risks.
As I mentioned earlier, we don't suggest that our salmon are going to feed the world. We do suggest that our tilapia and carp, which require the salmon to demonstrate the proof of concept and to build up a business, but the tilapia and carp, in fact, are going to be significant contributors to food security. They won't feed the world, but they contribute to food security. And every piece, every contribution that brings us closer to the point where you don't have to face famine, where you don't have to face starvation, in particular where you don't have to have 800,000 people, 800 million people a year going to bed hungry. I think that's important.
The truth is, the only value to genetically modified animals or plants is for the companies who own those patents. It doesn't make for better tasting food. It doesn't make for better production of food. It's really not good for the planet, but it really is good for the people who own those patents, who are trying to own our food. And the more that people know about this, the more resistant they will become. So lobbyists work as hard at prohibiting labeling as for anything else, because it's in their interest to keep the consumer ignorant.
Every time you walk into a fast food place, every time that you buy conventional vegetables, you and I am responsible for the pesticides being used, the incredible cruelty to these animals, the destruction of our forests and wildlife, and 70% of our endangered species are created through farming and ranching in the United States. We're complicit in those moral crimes, whether we know it or not. And so it's not just an environmental crisis, it's a moral crisis. And we're never going to solve that by being mere consumers. We have to say, no, we are creating either the solution or the problem.
As one of the few molecular biologists who are also skeptics, of course I've traveled quite a lot and given talks many places of the world. And the proponents of genetic engineering always jump on and say that a lot of the scientific arguments I use are exaggerated. But I have one particular argument that they never start discussing, and that is when I say that one of the main risk issues of genetic engineering is that 95% of all competent scientists in these fields are working for the producer side, and only 5% are really genuinely independent. They never discuss that, and that makes me suggest that maybe the situation is even worse, because I have no data for this. It's my own invention. The reason I mention it is, of course, that the day the percentages are 100 working for the industries and 0% that are really independent, then we have both a very serious scientific problem in society, but we also have a very, very serious democratic problem, as you may imagine and realize.
You're watching Free Speech TV.
It's another sign of the nation's weakening economy.
I take it as my task this evening to try to present a sense of what is going on in the American economy and around the world, since the United States plays such an enormous role in the world economy, to give you a sense of how we got into this situation and